Hi, my name is Avgoustou. I'm a recently graduated PhD student and a current postdoc at the Department of Oncology and Metabolism. So today I'm going to talk you a bit about my research. So the main aim of my research was the development of small molecule antagonists for the treatment of pancreatic cancer through targeting adrenaline 2 receptor. Pancreatic cancer, even though it's only the 11th most common cancer in the UK, the survival rates are quite low, with only 21% surviving for 5 years, and this is directly reflected by the mortality rates of the disease, which are significantly increased during the last few decades, and they are due to increase even more in the next few decades as well. The main treatments for the disease are chemotherapy or surgery. However, they are not able to increase significantly the lifespan and the quality of life of the patients. So this is showing the need of development of new targets and new therapies for the treatment of the disease. So adrenaline is one of those targets that our group is interested in. It's a multifunctional protein which acts through a G protein coupled receptor with which is belong to beta class G protein coupled receptors together with CGRP. So these receptors are formed by when the two different receptors are linked together, which are the CLR receptor, which is a 7 transmembrane domain, and the single transmembrane domain, which is called receptor associated modifying protein, which acts as accessory protein for the receptor. So it's determine their functionality and pharmacology. So when the CLR binds with the RAMP1, we have the development of the CGRP receptor, and when the RAMP2 and the RAMP3 binds with the CLR, we have the development of AM1 and AM2 receptors. So AM was shown previously to be involved in the regulation of blood pressure, and especially when acting through AM1 receptor. However, recent studies have shown that it's also linked with progression and the survival and the prognosis of several cancers, including pancreatic and breast cancer. And this is the main reason why our group was trying to target this receptor. Also, CGRP was shown to be involved previously in the progression and of pain. And that's why there are a lot of antagonists that have been developed recently or have been developed at the moment for the treatment of migraine. Using these receptors, using this rationalized and structure-based modeling system, we're able to develop our own homology model for the receptor, which was based on the AM1 and CGRP crystal structures found in the literature. And also using the already developed antagonists for CGRP, we're able to develop our own antagonists that will be more specific and more selective for the AM2 receptor. So this we were able to develop more than 1,000 compounds which were screened in vitro for against the different receptors. And after the screening in vitro, depending on the potency and selectivity, there were progress to more in vitro and in vivo assays. So after the pharmacological characterization, the compounds that were suitable for use in vivo based on their pharmacological characteristics, they were used for the treatment of pancreatic cancer xenograft models, when we have tumor cells injected subcutaneously in nude mice, and we're treating them with our antagonists to determine their functionality and potency. So what we've seen from our in vivo studies, we managed to decrease growth of the tumor development quite significantly, by more than 50%. And this was directly reflected on the immunohistochemistry analysis we done to our sections after the in vivo studies, which they shown a significantly decreased proliferation and angiogenesis in the tumors, and also we have increase in necrotic areas in the tumors as well. Another important characteristic that was shown in these tumors was the reduced the presence of fibrous tissues, which is one of the main characteristics of pancreatic cancers. Pancreatic tumors, which they develop these desmoplastic areas around the tumors, which make the treatment to be more difficult, because the drugs are not able to enter the tumor, so that they're not effective as we would like to be. So it's another possibility that treating with our antagonists will be able to decrease these dermoplastic tissues around the tumor, so we'll be able to target the tumor more effectively. So this project allowed me to work with a big group involved in a multidisciplinary team, and I was able to meet a lot of people in different groups and different disciplinaries. And also I was able to get my postdoc, which I'm currently working on the further development of these molecules. So we'd be able to get them into clinic and help patients with pancreatic cancer and also other cancer types that they're not treatable at the moment. Thank you.